In this presentation, we'll explain why you need to consider the purpose of your study when planning the experimental design. Depending on what you're trying to achieve, the correct choice of experimental design will not only improve the reliability of any scientific conclusions you make, but will also enable you to reduce the total number of animals used during your investigation. In this presentation, we will learn about the 3 stages of an experimental investigation: pilot studies, hypothesis generating experiments, and hypothesis confirming experiments. We will consider why you need to run these different types of experiment, how the experimental designs required varies depending on the stage of the whole study, and how you can reduce animal use by considering the 3-stage process as a whole. It is rare for us to perform a single one-off experiment in isolation. We usually need to run a series of experiments to investigate a scientific hypothesis. When you start planning an experiment, you need to consider what you're trying to achieve and how it relates to any previous experiments you have already performed. For example, perhaps you are setting up a new animal model and have no information about its reliability. Before testing any compounds using this animal model, you'll need to optimize the model parameters. Alternatively, you might have an established animal model, but you don't know how big the size of the response to the intervention may be. Finally, you may be able to predict the outcome of the intervention, but you need to confirm the prediction is reliable using an independent experiment to test the predefined hypothesis. Each of these scenarios requires a different experimental design. The size of the experiment is also influenced by the hypothesis you are assessing. Traditionally, we test for a difference, i.e. we are trying to confirm that the intervention has an effect compared to a control. However, this is not always the case. Perhaps instead you are trying to confirm that 2 interventions are equivalent, for instance when comparing a novel treatment with an active control. In this situation, you need to run an equivalence test, and we will focus on that issue in a separate resource. Sometimes you need to run an experiment to obtain an estimate of the background variability of the measure of interest. This estimate is needed in order to calculate an appropriate sample size for a future study. In such cases, you'll need a suitably large design so that the variability estimate is likely to be reliable and reproducible. While you need to think about all the above points when designing an experiment, it can be argued that there are different categories of experimental investigation, and these lead to different strategies for statistical analysis. Experiments can either be exploratory, where the purpose is to investigate whether or not there seems to be a response to the intervention, or hypothesis testing, where a specific and predefined hypothesis is being assessed. Each type of experiment requires different statistical analysis testing strategies. When investigating the response to an experimental intervention, you should begin by running exploratory assessments. These can be informal experiments, and you can be flexible about what you do when you assess the data. Perhaps you'll be running an experiment to identify the size of the effect or to identify a window of opportunity, for example when assessing the effect of drugs on a measure of interest. Any statistical analysis should aim to provide a reliable estimate of the size of the effect that you can expect to see in future experiments, for example the difference in group means. The statistical analysis should also give an idea of the reliability of these estimates. You can obtain a measure of the reliability of the estimate by estimating the variability of the response, such as the standard error of the mean or the confidence interval around the estimate. These can then be used when planning the more formal experiments designed to confirm predefined hypotheses. At this stage, it can be argued that the statistical analysis should be tailored to suit the questions raised once the data is generated. Perhaps an unexpected result is observed, and the statistical analysis is adapted to reflect this. Such strategies can result in false positives, but we argue such risks are acceptable at this stage, as long as these analyses are reported as tentative and not conclusive. Once the exploratory assessments have been performed, The investigation now moves on to testing hypotheses that have been generated during the earlier experiments. At this point, these experiments may test formal hypotheses and are performed to generate conclusions about whether the intervention causes a statistically significant change or not. The size of these experiments should also be planned in advance and be of appropriate size. Information gained in the exploratory experiments should be used to define the appropriate sample size using power analysis. We shall now describe a three-stage process that you should always consider when performing scientific investigations. The recommended experimental process provides a framework that should help you when planning your experiments. Although the process is not necessarily the same for all types of study or study objectives, the recommended experimental process involves three stages: the pilot study, the hypothesis generating experiment, and the hypothesis confirming experiment. The first two can be categorized as different types of exploratory assessments, whereas the third is a more formal hypothesis testing assessment. When you come to design your own experiments, you will need to consider what you are trying to achieve and see how it fits into this framework. Let us consider these three types of experiment in more detail. The pilot study. You will need to run one of these experiments if you do not know a great deal about the animal model you want to use, or perhaps you need to run a feasibility study to see if the animal model is likely to work. These are usually small experiments with small sample sizes per group. These designs are not intended to assess the effect of individual interventions, but to enable you to look at overall general effects, such as a preliminary assessment of a new animal model or treatment response. These designs should be seen as the first step in the process of developing the experimental procedure so that it can be used to test hypotheses. They can also be used to evaluate many different types of responses within a single experiment, for example body weight, food intake, basal metabolic rate, etc. At this stage, you might also want to optimize the animal model, as this will reduce the total animal use in future experiments. For example, you might want to investigate a range of factors that might influence the response, such as strain or sex of the animal you plan to use, or how long after dosing you should run the behavioural test, or perhaps how old the animals need to be. The use of factorial designs will enable you to investigate many experimental factors in a single experiment while not using too many animals. The hypothesis generating experiment. Once you have checked that the animal model is reliable and suitable for your needs, and have perhaps optimized the model in terms of the factors that affect the findings, you can now move on to the hypothesis generating experiments. At this stage, you should have an animal model that you can rely on and now be thinking about the effect of the intervention. You can't predict what the impact of your intervention is likely to be, and perhaps you don't know how variable your response is. Hence, you don't know how big a study needs to be to confirm your hypotheses are correct. These experiments are therefore still exploratory, where you are investigating the effect of the intervention. These experiments can be used to explore more than one type of response, in other words, to develop more than one hypothesis about the response to the intervention. It is important that the experiment is large enough to provide a reliable estimate of the variability of the measure of interests across the experimental units. We do not recommend performing statistical hypothesis tests on the data generated from these experiments, for example generating p-values, as you don't yet know what your sample size needs to be. These designs should, however, be large enough to generate a reliable estimate of the variability of the experimental unit. This estimate can then be used in a power analysis to calculate a suitable sample size for a future experiment that will enable you to test your hypotheses. The hypothesis confirming experiment. You can now move on to the formal hypothesis confirming experiment to independently test your hypotheses. These experiments should be used to confirm your hypothesis because you should believe you can now predict what the response to the intervention is. At this stage, you should have identified a suitable sample size. This is estimated using power analysis based on the biologically relevant effect that you expect to find and the variability of the response across the different groups within your animal model. As the experimental design is of a suitable size, you can now consider generating p-values and declaring statistical significance as part of the statistical analysis. You may also want to make adjustments to p-values using multiple comparison adjustment techniques. You should only report the results of the assessments of the hypotheses that you planned in advance. This shouldn't stop you interrogating your data for other unpredicted responses to the experimental intervention in a more exploratory way, but you should avoid the temptation of reporting such assessments as conclusive. These experiments might seem unnecessary and a waste of animals, but it is important to confirm that your hypotheses are reproducible in an independent, well-controlled and pre-planned experiment. You may also want to consider the internal and external validity of the hypothesis-confirming experiment. The internal validity is the extent to which the results of a given study are explained by the response to the experimental intervention, rather than some other unknown factor or factors. Examples of unknown factors could include the experimental environment, the animal strain, flaws in the design of the study, for example, the sample size was wrong, or the conduct of the study. For example, the study was not randomized or blinded. Internal validity involves standardizing the conduct and conditions of the experiment as much as possible to reduce variability arising from these unknown factors. The external validity is the extent to which the results of a given study are applicable to, or generalized to, other studies, study conditions, animal strains or species, especially humans. To improve external validity, you might decide to rerun the hypothesis-confirming experiment in a different lab, using a different supplier, in a different genotype of animals, or over a period of time with different technicians. Note the precautions taken to improve external validity are likely to diminish internal validity. While no single approach will work in all cases, we strongly recommend you aim to conduct a three-stage process when running your experimental investigations. Pilot studies should be small and serve to assess the feasibility of using the animal model. They should include as many experimental factors as possible to enable optimization of the factors of interest in future experiments. Once you have selected and confirmed the feasibility of the model, use hypothesis-generating experiments to explore your ideas. As they are more tentative and merely investigatory in nature, you are free to explore many different hypotheses and shouldn't worry about finding false positives at this stage. Such results will be confirmed or not in an independent follow-up experiment. For that reason, we strongly discourage you from reporting results from this type of experiment as definitive because there is a high risk of finding false positives. In fact, it is likely that the problem with the non-reproducibility of animal research stems in part from the reporting of results from this type of experiment as conclusive. These studies should also be large enough to obtain an estimate of the variability of the measure of interest in the animals being tested. Finally, plan to run a hypothesis-confirming experiment to confirm any hypotheses you have formed during these earlier experiments. You can have confidence in the conclusions because the experiment used an appropriate sample size. Now you've seen this presentation, you can go on and try out the exercise to explore these ideas further.